Good morning, everyone. Today, in this lecture talk, I'm discussing with you introduction to animal tissue culture. Okay, animal tissue culture refers to the culture of whole organs, tissue fragments, as well as dispersed cells on a suitable nutrient medium. Okay, and this can be divided into organ culture and cell culture. Okay, how are differentiating this one? Mainly based on the whether the tissue organization is retained or not. Okay, now let us go through the definition. What is the definition here? In vitro cultivation of organs, tissues and cells at defined temperature using an incubator and supplemented with a medium containing cell nutrients, okay, and other growth factors is collectively known as tissue culture. Make a note of this one because usually this question will be asked during your viva. Okay, there are different types of cells grown in culture, include connective tissue elements such as fibroblasts, skeletal tissue, cardiac epithelial tissue like liver, breast, skin, and kidney. Okay, and many different types of tumor cells. Now let us go through some of the terminologies. Okay, now let us understand the first one, primary cell culture. Okay, derived directly from the source animal and has not yet been subcultured. Okay, those are known as primary cell culture. It includes primary monkey kidney or monkey kidney cells, primary rabbit kidney cells, chick embryo fibroblast, human amnion cell culture. All this which comes under primary cell culture. And next one is finite cell lines. These cell lines are capable of limited number of passages before undergoing senescence, or these cell lines die after several subcultures. Okay, that includes human fibroblast cell cultures such as MRC5 and WI38 cells. Okay, further here continuous or transformed cell lines. Okay, these cell lines are immortalized cells that can be pass aged without limit. Okay, or cell lines that can be maintained indefinitely. Okay, for example, HEP2, that is human epithelioma of larynx, or HeLa, then A549 or Medrin Darby Kenin kidney cells. Okay, frequently asked question is this one, transformed cell lines. Okay, next one, hematopoietic cells. These hematopoietic cells, such as peripheral blood mononuclear cells or umbilical cord mononuclear cells, are required for the growth of viruses such as EBV, then HHV6, 8, etc. Okay, so that is not HIV, that is the HHV. Now let us go through about the cell culture. Cell culture is the process of isolating cells from an organism and growing them in a scientifically controlled environment. Okay, hope you understand here about the cell culture. What is cell culture? Cell culture is the process of isolating cells from an organism and growing them in scientifically controlled environment. Okay, today cell lines derived from animals including humans are grown and used for research in laboratories around the world, right? These cells can continuously divide, okay, in a given the right growth conditions. The first of these cell lines to be established is known as HeLa cells, HeLa. This is a cell line established from a young African American woman named Henrietta Lacks, okay, who died of an aggressive cervical adenocarcinoma in the year 1951. Okay, she presented to the gynecology department of Johns Hopkins Hospital in Baltimore, Maryland, where they found the tumor on her cervix. When a biopsy was taken from the tumor, okay, one specimen was given to George Gay and his colleagues at the tissue culture laboratory at Johns Hopkins, who had ambitions to isolate and maintain normal and diseased tissue as temporary or stable organoids are as derived cell strains. Okay, usually during viva, if they ask what are these HeLa cells, what is this HeLa cells, that time we can give this particular information. Okay, keep this in your mind about the HeLa cells. Put one star mark. HeLa cells is very important in this particular slide. Further, if you want to elaborate about HeLa, here is the answer. Okay, HeLa is an immortal cell line used in scientific research. Okay, it is the oldest and most commonly used human cell line. The line is derived from cervical cancer cells taken on February 8, 1951 from Henrietta Lacks, a 31-year-old African American mother of five who died of cancer on October 4th during the year 1951. Okay, HeLa cells have been used to test the effects of radiation, cosmetics, toxins, and other chemicals on human cells. Okay, they have been instrumental in gene mapping and studying human diseases, especially cancer. So, students, kindly go through about HeLa cell lines. You can write this particular information to paragraph here, and in this particular both the paragraph you can write for the HeLa cells. Okay, usually this will be asked for 2 marks or 1 marks, and during viva also they may ask what is this HeLa cells. You must be able to answer this. Okay, now let us move on to the history. In the year 1885, Wilhelm Rox removed a section of the medullary plate of an embryonic chicken and maintained it in a warm saline solution for several days. Okay, and he established the basic principles of tissue culture. Okay, in the year 1907, the zoologist Ross Gravely Harrison demonstrated the growth of frog embryonic cells that would give rise to nerve cells in a medium of clotted lymph. Okay, whereas during the year 1903, Joly studied the behavior of animal tissue explants immersed in serum, lymph, or ascites fluid. Now let us go through applications of animal cell culture. Okay, what are the applications here? The main application here, they play a very important role in production of antiviral vaccines, which requires the standardization of cell lines for multiplication and assay of viruses. Okay, then this play also very important role in cancer research, which requires the study of uncontrolled cell division in cultures. Further, they play a very important role in production of monoclonal antibodies, production of pharmaceutical drugs using cell lines as bioreactors. Okay, and also chromosome analysis of cells derived from womb is also possible. Further, tissue engineering use of artificial skin and bones can also be seen as the applications of animal cell culture. Study the function of nerve cells and signaling in neurological research also play a very important role. Okay, then to study the effect of toxins and pollutants effect related to stress, it is also one of the applications. Okay, then finally here, genetic manipulation can also be used. This animal cell culture, this that will be easy to carry out in cells organ culture. Now let us go through here medium. Okay, medium here, formulation of consisting of various nutrients used for culture of animal or plant cells or tissues, organs or microorganisms. organisms, etc., that can be done through the medium or culture medium. Okay. So in the culture medium, we have two types of media, right? One is the natural media, other one is the artificial media, or natural medium or artificial medium. So what is this natural media or medium? This natural media is a biological fluids used as a medium. Okay. Whereas artificial medium, with the medium having various organic and inorganic nutrients, they and may or may not contain a serum, etc. Okay. Serum containing medium, that is artificial medium, supplemented with 2 to 20% serum. So what is serum here? Liquid part of the blood, which is straw yellow colored, is known as the serum. Serum-free medium can also be used. This is also one of the artificial medium lacking serum. Okay. Instead, with the presence of growth factor hormones, other proteins, etc., may be added as per need or requirement. Next one is chemically defined artificial medium. Okay. Without serum protein. Okay, one is chemically defined. Chemical defined is the artificial medium without serum. Further, protein-free media. This is also one of the artificial medium free from any proteins. Now let us go through basic requirements for animal cell culture. Among the essential requirements for animal cell culture are special incubators to maintain the levels of oxygen, carbon dioxide, temperature, humidity as present in the animal body. Okay, the synthetic media are chemically defined media with vitamins, amino acids, and fetal cough serum can be seen here. In that, first one is basic important, that is temperature. The most important one, this particular temperature. Okay, most of the mammalian cell cultures, the temperature during cell culture, the temperature which is maintained at 37 degrees Celsius in the incubator, as the body temperature of human is 37 degree Celsius. Okay. The next major requirement is culture media. The culture media is prepared in such a way that it should provide the optimum conditions of factors like pH, osmotic pressure, etc. Okay. Next, the most important one is the pH. Most media contain or maintain the pH between 7 to 7.4. Okay. pH is very much essential to maintain the proper ion balance, optimal functioning of cellular enzymes, and binding of hormones and growth factors to cell surface receptors in the cell cultures. Okay. The regulation of pH is done using a variety of buffering systems. Okay. So in this case, these are the some of the basic requirements of animal cell culture. Okay. What are those? Temperature, culture media, pH. Okay. Further, you can also recall about this osmolality. Okay. You can put a star mark, 1 mark or 2 marks. Usually this question will be asked. Okay. You must be able to answer. Osmolality refers to the concentration of dissolved particles of chemicals and minerals, okay, such as sodium and other electrolytes in your serum. Higher osmolality means you have more particles in your serum. Lower osmolality means the particles are more diluted. Okay. So what is this osmolality? Osmolality, that is nothing but there you can see the presence of salt, glucose, and amino acids in the growth media, which determines the osmolality of the medium. Okay. It should contain chemical constituents which the cells or tissues are incapable of synthesizing. Okay. Generally, the media is a mixture of inorganic salts and other nutrients capable of sustaining cells in the culture, such as amino acids, fatty acids, sugars, ions, trace elements, vitamins, cofactors, and ions. Okay, glucose is added as energy source with the varying concentration depending on the requirement. Okay, and here Phenol red is added as pH indicator of the medium. Okay. Hope you understood today's introduction to animal tissue culture. I, Dr. Pratibha K, signing out for today. I'll be back with some more videos. Stay tuned with me. Thank you for watching.